All right. Hello, everyone. So I'm Meredith Ashby. I'm part of the marketing team here at Element, and I want to welcome you all to the Element webinar series, where today's topic is accelerating your science with DNA sequencing designed for you. First, a couple brief housekeeping announcements. The presentation portion of our webinar will be followed by a Q&A session, and you're welcome to submit your questions for either speaker at any time by clicking on the Q&A icon in your Zoom webinar control panel, which you'll notice is distinct from the chat icon. We will be recording this webinar and making it available for reviewing in the next few days, so look for an email with the link to the recording. With that, let me introduce our first speaker, Matt Kellinger, co-founder and VP of Biochemistry here at Element, who will be talking to us about Element's forthcoming Cloudbreak chemistry. Matt brings a wealth of biotechnology experience to Element. Prior to co-founding the company, he served as a staff scientist at Illumina, working on polymerase engineering and characterization of multi-enzyme systems to support and develop the future of genome sequencing enzymes. He has a PhD in cell and molecular biology from the University of Texas, where he employed pre-steady state kinetic characterization and global analysis of fitting of HIV reverse transcriptase to elucidate drug resistance mechanisms. And in his free time, Matt enjoys thinking about science. Matt, take it away.
Thank you, Meredith. Sorry to have to make you read all of that as an intro. Good morning, everyone. I'm Matt Kellinger. I'm a co-founder of Element Biosciences and the VP of Biochemistry. And today I'm here with you to share some of the exciting work we've been doing to develop new sequencing chemistry that will be releasing in April on the AVITI platform. And I had the pleasure of sharing these slides recently at AGBT. We thought it would be great to sort of expand the audience list and let everyone see what we're working on and what's coming from Element. Very briefly, we launched the AVITI sequencer last summer with this idea of have it all with AVITI, really producing a desktop sequencer that's an unmatched combination of performance, cost, and flexibility. And some of the initial specs that came out on the system, 90% Q30, high output in terms of 2 flow cells generating 600 gigabases of data. And really, the costs that come with that around $1 per million reads of doing counting applications and $5 per G for human build applications, things like that at 2x150. One of the big advantages of the system is 2 independent flow cells. So the instrument has 2 sides that can run independently but concurrently, which effectively provide 2 instruments worth of flexibility in just one system. If I go forward a little bit now with the instrument, on the market, what we've seen is that genomics is really a large umbrella for a lot of different types of applications and different markets. We've seen the AVITI get adopted across large genome centers, in core labs, in the biotech industry, with clinical partners, in government agencies, and in commercial testing labs. And I want to thank the customers who share their name to this slide. It's great to get to highlight some of the work they're doing and how they're using the AVITI platform to accelerate that work. So if we look about field performance and really how the installation process goes, I think the team here at Element is very committed to making that one a smooth process. From the minute we receive the order to getting customer up and running with sequencing is of the highest priority. So what we're seeing is that installations largely go very smoothly. The instruments get up running quickly so that they can facilitate collecting data and you can get started with the science part of what you're focused on. And if we look at the data quality that's produced when that science begins, what we're really seeing is meeting our specs, right? So the spec is 90% Q30 versus the 85% on the higher Illumina platforms. We do see some advantages on the platform that have become even stronger as customers have started to experience them, being the low optical duplicate rate, attributed largely to how we're amplifying your library on our surface, negligible index hopping, because again of that amplification technology we've developed, and then really strong performance post-homopolymer, and I have some slides on it a little bit later in the deck to show that what happens when AVITI chemistry encounters a homopolymer in the genome, and the performance post the homopolymer is really a striking difference. I think it's worth to take a minute to step back and sort of again introduce this idea of avidity sequencing, why it's different and the advantages that it brings. What we've done with this chemistry is we've separated the detection and calling of a base from the physical stepping to the next base in the sequence, and that comes with some advantages. First, we're able to tailor those biochemical reactions, which are quite different, to their specific functions. And in the detection part, those first 4 panels I show on the left, we're using substrates called avidites. Now, these are multivalent substrates, which really allows for the polymerase interacting with the polymer on the surface to do its job, lend high discrimination, and ultimately have a very strong signal for the eventual base call that's being made. After that, we can remove the avidite. So effectively what we've done is we've interrogated the base in question and made a base call without modifying the colony DNA in any way. This lets us then step to the next base, and a difference here is that we don't have scars that are generated in this process. So we're generating natural DNA, which again helps with error rates as we go through this cycle 150 times. In the lower left is a schematic of just one of those avidite molecules interacting with the poly. And again, what you see is avidity on display there. So multiple polymerases, each grabbing one of the nucleotides contained on that avidite, really lends that high specificity to the correct base call. There's a good stringency there. I do have a large QR code there to a link to much more detail on how avidity chemistry works in a preprint that we put on bioRxiv earlier this year. So what's coming this year, right? In April, we're going to announce, or in April, Cloudbreak soft biochemistry is going to be released. And the advantage of this chemistry is really where we focused our efforts in the last 9 months since the launch of AVITI. We made 2 promises when we started shipping AVITIs. One is that innovation would be continual. So we want to be able to use the same platform and reach greater and greater heights in terms of the biochemistry that's on that platform. We felt that the chemistry was in its early stages and had tons of headroom for improvement. And that's what we've done with Cloudbreak. The second commitment there really was to get a lot of feedback from customers on what they're interested in and the directions they want to go with their science. And I'll have more on that a little bit later in the talk. So what Cloudbreak offers, largely through 2 new enzymes used in that avidity chemistry cycle, as well as some advances in the engineering side on how we image and how we do that faster, is really a 10-hour reduction in overall runtime for 2x150. So we've saved about 20% of the runtime, going from 48 hours down to 38 with this chemistry. We maintain the same high data quality with our spec of 90% Q30, although most of the data is considerably higher quality than that. And we've made some advantages to the workflow itself. Again, sort of based on feedback from customers, we felt like we had the quality in the sequencing performance to put indexing at the beginning. This has the benefit of allowing users to generate an early report during the sequencing run in about the first 5 hours that gives you all the demux statistics, so you can see how your library pooling went and to see what bins the buckets are falling into from the index side. Also, for our Elevate library workflow, we've changed the process where you no longer need circular DNA library as an input. We've enabled the onboard circularization, taking that work out of the hands of library prep, putting it onto the AVITI at no additional time penalty. The instrument still is extremely flexible in terms of tuning its output to what's needed for experiments, and I'll go through and highlight that a little bit. And then we've increased the shelf life of the reagent consumables from 6 to 12 months. So twofold improvement there to ease inventory management. Go look at a schematic of the runtime. Again, here you can see those changes. So in the light blue is the change to indexing. So early in the run, we generated an index report. And then on the back end of the run, you're saving 10 hours. And where we found this to be extremely effective is starting runs every other day on the platform. This becomes a much easier task when the runs are done earlier in the day. The 38-hour runtime is a convenient interval to be able to start multiple runs every second day on either side of the instrument. Of course, the 38-hour performance for 2x150 is sort of the top line performance. If you look at other configurations in terms of either output or read length, in some cases for the 2x75s or more counting focused applications, the system can run with lower output in as little as 18 hours, if that's a critical factor in the type of data you have to generate, how fast it can be generated. And as I talk about on flow cell circularization, I want to just remind everyone of two workflows that really bring you into the AVITI ecosystem. The first is what we call the Adept workflow. This is for existing third-party library kits that generate libraries compatible with the Illumina platform. We have a step that still remains in library prep for the Adept workflow that circularizes those libraries so that circular DNA can then go into the sequencer and work with our amplification chemistry. What's different with Cloudbreak and the Elevate workflow is you still use the third-party kit, but you can get adapters and indexes from us that allow you to skip that circularization step, load linear library directly onto the system, saving you time in library prep. And here's a little bit more detail about how that process works. So, again, it's a commercial kit that you're used to using with a swap of adapters from us. We have many app notes and protocols available online how to do that, but it doesn't fundamentally change the workflow supplied by that third-party kit. And then that linear library can be loaded on the AVITI cartridge where it's brought into the instrument. The circularization happens on the flow cell, followed by amplification, and then ultimately into sequencing. So quality is always something that's extremely important in any sequencing application we're going to put out. You can see the V1 performance of the AVITI there in light blue, and we've done a lot of benchmarking against both NovaSeq 6000 and the NextSeq 2000. And the V1 chemistry, very strong performance in terms of higher quality than that competitor platform. And what Cloudbreak brings is more consistency in terms of the performance. So you can see the large gains that were made in read-to quality, and a large fraction of the bases being actually above Q40. This is all done at 20% faster times and with those simpler workflows. So a major way that we benchmark here is against human genomes. So, we're doing PCR-free library prep and then being very careful to analyze the data using downstream analysis software, doing full human builds, and then looking at where mistakes are made. You can see with the AVITI V1 chemistry and Cloudbreak, very similar performance in both SNP and indel calls for these full human builds of HG002. This benchmarks really well against data off the Illumina platforms, although one does have to be careful to maintain the differences that come from libraries. So library prep itself can add a lot of variation in these numbers. That's largely why we're showing the V1 to V2 performance. And if we look at GC bias for a build of a full human genome, again, you can see the Cloudbreak software with the AVITI V1, or the Cloudbreak biochemistry with the AVITI first generation biochemistry against, again, benchmarked against NovaSeq and NextSeq. What you can see is where a majority of the SNPs lay sort of between that 25 to 75% window of GC. 99.2% of the SNPs are there. You see very consistent performance in terms of SNP F1 score across that region, with slight improvements to Cloudbreak on the extreme ends of GC, where there's not much data, but performance still matters. Next to homopolymers, I found this very interesting when we first, the bioinformatics team here at Element first started to dig into this, and how I can orient you is on the panel on the left. If you're looking at the extreme negative numbers down towards the origin of that plot, that's basically upstream of a homopolymer as you're sequencing along. You can see both the Illumina chemistry and the element chemistry perform very well in those regions. The interesting thing happens when you get to position 0, which is the first base after a homopolymer. On this plot, you can see because of the avidity chemistry, and I think this is due largely to separating the detection from the stepping and how cleanly we can step through homopolymers, you're maintaining very low error rates post-homopolymer, whereas with traditional SBS, you tend to see very large increases in error after those sequences. And we looked at certain instances of these homopolymers, and in the end, we decided to look at all homopolymers in the human genome. So you can see on the panel on the right, there's actually a difference plot of all homopolymers in the human genome. 0 being performance is the same across the two platforms. To the left in blue is where Element fares better over all of the homopolymers in the human genome. And you can see that it's a vast majority of them where performance is very strong. If we look at some other applications, here's some data on exome performance with twist panels, which you can see in this plot is on the left. Above 0 is effectively, do you make the right SNP and indel calls, right? I think both technologies, the AVITI and the Illumina platforms, at the end of the day, make the correct calls. The large difference being in the quality of the data that comes off the Element platform leads to less candidate calls, specifically in SNP false positives that have to be weeded through to be able to find the correct answer. And then on the right is a pan-cancer panel looking at percent recall as a function of coverage depth. And what we find is that the AVITI sequencer consistently outperforms Illumina data at various coverage depths. And largely this is due to lower duplication, so less redundant data needing to be thrown away prior to analysis, and then the overall higher sequencing quality to be able to find the right answer. I think Sean is going to talk about this more in detail a little bit later on, but another interesting finding on the AVITI platform is we started to study what impact does insert size for the library have on our ability to look at full human builds. The technology that we put into AVITI, specifically how we amplify the DNA on the surface, is very conducive to increasing that insert size. It has less of a bias there in terms of selecting for small library fragments over large library fragments. The way this experiment was done was generating libraries of various insert size, sequencing them, and then on the left, looking at the aligned insert length as a distribution. And you can see those different libraries. The most interesting one on that plot probably being that brown color, which has a median aligned insert size of over 1,000 bases, with some library colonies generated over 2,000 bases for their inserts. And initially we're still doing 2x150 sequencing here, but the space between those two reads is getting larger and larger as the insert size gets bigger. That drastically helps with alignment. We're able to see on that panel on the right a strong NovaSeq run in HG002 in terms of total errors generated, and the AVITI platform with standard insert sizes next to it. When you go to long insert sizes, you see about a 25% improvement overall. This is a very interesting direction for us as we designed the amplification chemistry in AVITI to be able to support longer inserts. And I think there's clear advantages here when doing human builds, even if you're still at 2x150 read lengths. So I'd like to look a little bit past Cloudbreak chemistry, and really this goes to that second promise we made as we brought the system to the market, was that we wanted to interact with potential customers and current customers about what was needed in the space and what they were interested in the future. And 2x300 kept coming up as something that it would be great to have with the economics of the AVITI platform. It's enabling for certain applications, and it's something that's currently in research and development here at Element that will be releasing later this year in Q3. I'd like to provide an early look into the progress, and what you can see is already strong performance in terms of read 1 and read 2 over 600 cycles. We believe this is enabling for many applications today. And a key difference between the platforms that you can run the 2x300 application on is going to be in the number of reads. So on the Element platform, we're generating 400 million paired end reads at these error rates today, versus vast less number of paired end reads on the MySeq and NextSeq platforms. Also, a little bit of a plug, we're actively recruiting early access testing for this around summertime. I ask if you're interested in this, and this is an application that you run, please reach out either at our website or you can email that email address shown there. That comes directly to me. I'm monitoring it, and we're looking for potential partners to do early access testing. The last part of my talk today is really going to look at the economics of using an AVITI sequencer and some of the flexibility that the system performs. We designed the system to span a large range of outputs so that it be the right sequencer for many different applications and labs, knowing that the industry itself is very diverse. We look at how the AVITI sequencer stacks up against the NextSeq 2000. We put a calculator on our website. I think this is a really interesting tool that can be configured and played with. What we found is that even with modest usage, you can save $1.7 million in 3 years using an AVITI sequencer versus a NextSeq 2000. as we've seen in the field from Illumina, discounting of both systems and reagents. We did a comparison there on the right, even with the free NextSeq 2000, because the operating costs, the run costs and reagents are so much lower with the AVITI, it takes less than 6 months to break even and come out ahead. So I think that's a really, strong case for how the economics of the AVITI can benefit your lab. You can think about what could be done with that extra $1.7 million of research money. But that's not the only place where the flexibility stacks up well against competitor platforms that are available. If we look at AVITI versus the MiSeq, you can see that on the left, if you put the instrument in a lower number read mode, it'll generate that data faster. You're still able to generate about 10 times the number of data, 125 million paired end reads, in roughly the same time, a little bit slower, 25 hours versus 24. You're getting higher data quality, and your cost per G is significantly lower than the low output MiSeq system. If we start to get further into the desktop space, if we look at the HiSeq 550, here AVITI is put in the 500 million read output. You see 500 million reads versus 400 million reads with comparable runtimes, again, 2 hours slower at 31 versus 29. AVITI wins again on data quality, significantly higher here. And the push-go cost, right? That costs to start the sequencer of 1680, significantly different than around the 5,000 of the NextSeq 550, bringing in a cost per G at about 4 times less to generate even more data. If we look at the AVITI system against the NextSeq 2000 system, now is where the Cloudbreak chemistry, I think, really has advantages. The system in the full 1 billion reads output for one side is generating a little less than the 1.2 billion on the NextSeq 2000 platform. That runtime is coming in at 10 hours faster, again with higher quality. And the single price reagent cost of 1680 is significantly lower to push go, leading to an almost 3x reduction in cost per G. And we've got a path down to even lower cost per G values, which I'll talk about in a minute. If we look at AVITI against the NovaSeq 6000, the SP flow cell, here AVITI is generating more data on a single side of the instrument at a trade-off for runtime, the NovaSeq coming in at 25 hours and AVITI taking 38. Again, the quality is higher, with 90% of the data being Q30 or above, most of the data in the Q40 buckets. Again, cost to push go lower, and now we're at a cost per G that's still 2x lower than the NovaSeq platform. And if we look at the S2 flow cell, we can use the flexibility of the AVITI system to run both sides. So now we're generating 2 billion paired end reads at a comparable time of 38 hours versus 36. Again, clear wins on data quality, cost to push go, and overall cost per G. The last point I want to share with you all on the economics of the AVITI platform comes in something we announced earlier this year, the $200 genome, and how this can be done on the AVITI platform and the advantages that it offers. So the commitment here from customers is a commitment to a certain number of runs and a volume. And what we find with this commitment comes a $200 genome, but it's not just limited to whole genome sequencing. In fact, there's no limitations on how the sequencing kits can be used. You can use the kits to get a $13 exome. That brings the economics down to $2 per G, and for counting applications down to 60 cents per million reads. This is available today, and one other point I'll add about this is the lack of location constraints. So we've made it so that organizations that are not entirely centralized, right? If there's a site in the Bay Area and there's a site in Boston, they can work together to make the economics of this offering work. Both of those systems, even though they're not in the same location, can count towards the volume requirements. So if you are at sites with multiple locations, this might be a great option to be able to spread the AVITIs out, have sequencing on site, and be able to tap into the lowest costs today for genome sequencing. I think I conclude there. I can pass it back to Meredith.
All right. Thanks so much, Matt. That was a really terrific talk. And our next speaker is going to be Sean Levy, our CSO and SVP of Applications and Scientific Affairs. Prior to joining Element, Sean was the CSO at Discovery Life Sciences and a faculty investigator at Hudson Alpha Institute for Biotechnology. While there, he developed a comprehensive genomics facility that has supported more than 5,200 projects from locations around the world. Before Hudson Alpha, Sean was a faculty member at Vanderbilt University Medical Center in the Department of Biomedical Informatics and the Department of Molecular Physiology and Biophysics. He is the author or co-author on more than 200 peer-reviewed publications with over 45,000 citations. Sean is going to be talking to us about how Element technology that Matt shared translates into application improvements at a practical level for genomic scientists.
Thanks, Meredith. Thanks for the very kind introduction. Let me share my screen. All right, so I'm going to take an opportunity or take advantage of where Matt left off, and also take an opportunity to maybe re-emphasize a couple of the points, but also wanted to begin my section of the talk of calling out in very brief detail some of the very unique features of Element, and I'll expand on a few of these, really to indicate where some of the innovations have taken place. So Matt talked a bit about the sequencing chemistries. I'll mention a few more details there as well. But we really wanted to take a moment to say that the platform really from start to finish, from the workflows through the flow cell to the sequencing chemistry, as I kind of proceed around the center oval in a clockwise fashion, to the instrument, to even the way we do base calling and from an accuracy and throughput perspective, has all been very much reimagined with Element, and as Matt mentioned, we'll continue to innovate at that core. Although there's been a lot of innovation on the sequencing aspects of it, there was one very clear recognition we wanted to make sure we call out in this webinar, and that is that the genomics ecosystem has been contributed to by many commercial, academic, and groups around the world to really give us the diversity of applications that we see today. And so when AVITI was designed from the ground up, it was to embrace that ecosystem both on the front end and on the back end, and that's represented here. Shown on the left-hand side under the prepare is a number of companies and technologies that we've worked with over the last year or so. Many of these are detailed on the website with a large number of app notes, providing some more specifics around what that relationship is and what we've brought forward. But the bottom line is the sequencer was made to be able to drop into an existing laboratory, either as a replacement for an existing platform or to complement an existing platform, without the requirement of having to change your upfront library prep workflows or even your downstream bioinformatics. And in fact, many of the benchmarks that you've seen Matt show and some of the ones I'll show, the only variable was the sequencing platform. By having this compatibility, we're able to take precisely the same library material, sequence it on one platform, convert it for our sequencer via the Adept protocol, and then sequence it on our platform, and then also hold all of the downstream bioinformatics processes exactly the same. Now, there are some advantages with some of the partners we've used by using some element-specific workflows where you can get a bit higher quality results or perhaps a bit better resolution, but they're not a requirement. So this does make benchmarking and compatibility with existing workflows very, very convenient. Now, all of that said, we are, of course, not slowing down on some of the innovation. Some of these things are I'll talk in more detail with, but we also wanted to call out that the AVITI OS system is the operating system that runs the sequencer is a fully integrated platform operating system. We control all aspects of the firmware and software that runs on the platform. We can update over the air. It doesn't require engineers to be on site. Of course, those updates are done with the full acknowledgement of the local user, meaning there's no surprise updates for obvious reasons. In many laboratories, we need to very carefully control change logs. But when an update is accepted, there will not be a need for most updates for an engineer to be on site. Our senior vice president for software and informatics, Francisco Garcia, gave a brief talk at AGBT detailing Element Bio Cloud, which gives you a sort of a worldwide view of instruments within an organization to allow very substantial instrument monitoring, run setup, telemetry, et cetera, as well as configuration changes. So this is not a cloud platform where we would require you to use our infrastructure or a specific cloud infrastructure to do your data analysis and storage. Quite the opposite. This is a system that is designed to make your choice of downstream computing and downstream storage very convenient to use and be able to do so in a federated manner so that data security is at the forefront. But you're not tied to a single storage architecture or a single cloud provider at all, as indicated on the bottom. So, we're very committed to software integration of generating a sample to sequencing to results type of workflow. And then some of the things on the left-hand side of the slide, I'll go into a bit more detail in a few moments. So Matt described this in his part of the talk. What I just wanted to emphasize is shown on the bottom panels. And this is and the reason for the emphasis is this is such a key difference between sequencing by synthesis chemistries in the past, as well as chemistries that are now being used by a number of new platforms that have come up. It's very exciting in the field to have choice and to have options and be able to tailor those options of where they're most advantageous to either a throughput or an application. But I think it's also very important to call out where they are different to allow that to be a very comprehensive decision. Because there are effects in the downstream data due to these differences. So working from the lower part of the slide from left to right, we begin with a library molecule represented as a circle where we use a rolling circle amplification. As you see, as in the next, where we have this linear copy of showing multiple copies of the same DNA fragment that are now linear. There is a unique compaction event that takes place to make a very compact polyny, which is then shown in the cartoon on the far right-hand side. Important other call out is given this is a rolling circle amplification, we don't make copies of copies. So this is we make multiple copies of the same original template, which allows us to avoid the propagation of clonal error as we go through the amplification process. And then as Matt pointed out, the multivalent interaction of the avidite molecule with those copies of DNA is another critical difference in our sequencing chemistry. And of course, finally, the separation of the binding of the avidite molecule for its detection of the base. And again, to reiterate what Matt said, this is a 4-color system. So each base is fluorescently labeled. There are no dark bases. Is separate from the stepping process where we use a natural nucleotide that's only reversibly terminated. So we have no scarring on the DNA, as Matt talked about briefly before. You saw the QR code indicating the sequencing by AVITI paper that we've published on bioRxiv. There's a couple of other papers that are up there now as well. One of them is detailing low-pass sequencing using imputation in a collaboration with GenCove. And then in collaboration with some colleagues at UCSD, in medRxiv, there's a publication showing the use of avidity sequencing and whole genome sequence in whole genomes from retinal degeneration pedigrees to identify causal variants. And so those are all currently available, and we have more coming. We're very proud of our data quality. This is shown, here's some results of our quality score recalibration. The call out here is these are PCR-free libraries. Stated simply, as we evolve sequencing technology and as Element has advanced its ability to generate quality DNA, we can now clearly differentiate quality challenges that may come from either sample quality, library preparation methods that are used, and clearly in our hands, PCR-free libraries where you don't have those additional amplification steps taking place lead to the highest quality data as you see here. Also important to show that our training versus testing is very accurate across the range of Q-scores. And we also make these data sets as well as the base recalibration command lines that are used using open source tools to create these entirely publicly available so that the our interpretation and in creation of these training tables can be completely validated by anyone who so chooses to look at that work. Matt mentioned this on the homopolymer error. I won't belabor it other than to say this is a significant change that we've observed that we're quite interested to continue to pursue. And here is just a little bit more granularity on some of this is if you look at as a function of homopolymer length. So the results that Matt showed as far as the error rate were really on the longer homopolymer side, 11 nucleotides or longer. Here is a distribution of how that looks as we step up on a single base by single base increase in length. So you clearly see that the longer the homopolymer, the more challenged some other chemistries can be in terms of sequencing post-homopolymer. Of course, the key question is, why does it matter? We have a publication underway with some collaborators where because of this ability through homopolymers and related sites, it gives us some enhancement of indel sensitivity. And it also is, we've been seeing consistently that we see higher coverage of MSI informative sites in oncology samples. So these observations do matter in certain contexts, and we're continually exploring as to how else they matter. And I'll show you some examples from an assembly perspective at the end of, again, why these quality differences actually make a difference. A couple more comments on exome sequencing. This is a little bit of detail of what Matt summarized in one of his slides around our work with Twist. We included some content from both their cancer panel as well as their exome design, and then really handled these samples, or these libraries were prepared to allow a direct comparison to competing technologies, as if they were going onto an Illumina platform, given that it's the most convenient platform that as far as what Twist has available. So a very, very high quality base calling, as you see here. Our base call accuracy was actually 97% Q30 and 87% Q40 across the total cycles that were used. That's summarized on this one. And if we look at some of the typical statistics that are used in exome analysis, fold 80 penalty, duplication rate, and on-target rate, as well as looked across replicates, we wanted to, that's summarized here. So a little bit, for those of you not interested in exome sequencing, this is not a particularly exciting slide, but I wanted to call out one is a very consistent fold 80 penalty of below 1.4, given that showing that the uniformity of coverage is strong. Our on-target rates are very high at greater than 90%. And then probably most significant is the duplication rate. We have some additional studies, we're happy to share the data, where we've taken similar samples and sequenced them very, very deeply on the AVITI technology. I think most assumptions are that exome analysis, as you get to several hundred X to maybe even approaching 1,000X coverage, that your duplication rates become very unmanageable. What we found is there is perhaps for many samples an overestimate of the percentage of duplicates that come from PCR versus that come from optical duplication on the sequencing platform. Because as you see here, we're maintaining, in this case, this is a 100X exome, we're maintaining duplication rates at below 2%. And in fact, we have examples of data at greater than 2,000X coverage in exomes where our duplication rates remain comparatively very low, in things in the teens to low 20% range, allowing very, very deep coverage of exome analysis. So here's a little bit more housekeeping in terms of the variant calling metrics. You can see as far as SNP and endel, recall, precision, and F1, everything looks very good across 2 benchmark genomes that are available from Genome in a Bottle. And this was an evaluation was done using HAPI to the 4.2.1 truth sets in both cases. You can see the depth of coverage of 72 and a little over 90% respectively for the two samples. So it looks very, very good. Matt alluded to some of the long insert whole genome sequencing, and I wanted to expand on that just a little bit. So we did some simulation, and this is a few of the details on that simulation data. We used NEAT, which is I provided a reference with the DOI there. It's available on GitHub. Anyone who is interested in replicating this, please reach out to us. There's a couple of caveats of using NEAT that we can save you a lot of time in working through it, but it's a great tool to have in the field. We wanted to use publicly available open source tools as much as possible to make this happen. So essentially what we did with this simulation was simulated if the sequencing data is perfect, meaning that there is no sequencing error introduced on a library prep or a sequencing platform, and then started manipulating the data from there. Excuse me. So here you see, if you hold everything else constant, the effect of using a reference genome that, of course, you're aligning to, and then through the NEAT process, we introduced variants that would recapitulate, or from a VCF file that would allow you to simulate a typical Eastern European descent genome, about 3.5 million variants being introduced. And then we started just simply changing insert size. So all the variants are the same, all the samples are the same, all the data is the same. The only variance now is we change the insert size during the simulation, and you can see the effect on error. So starting with 300 base pair, and this is all paired end runs, and then increasing to a 1,500 base pair error, you can see you can almost cut the amount of total error in the system by about half, with the greatest effect being a decrease in the SNP false negative rate. And you can see, smaller effects in some of the other error classes as you see on the slide. So the reason this is important is that what it means is that simply by increasing insert size in a library in a genome as complex as a human genome, you can gain quite a bit of performance in SNP discovery or SNP detection. If we also ask the question a slightly different way, and let's take a 2x150 read condition at a 500 base pair insert and call that like a truth set, meaning set that to 0, and then ask what is the relative change in error as we manipulate not only the insert size, but now also the sequencing conditions. And what you see very simply is for single end technologies, you need read lengths at 500 base pairs or greater to come close to the performance of a 2x150 500 base pair insert. Not represented on the slide is going above 500 base pair inserts. You saw on the previous slide that there is substantial gain there. And that was just mainly, we already knew that at a 500 base pair insert, paired end sequencing is vastly outperforming single end or shorter insert sequencings. But we just really wanted to get a comprehensive benchmark on this. But interestingly, if you look at a 1x200 base pair read, you can see we're introducing almost 37,000 errors into the data set for no other reason other than just the inability to uniquely align those fragments and then accurately call variants in this process. So insert size matters, sequencing paired end capabilities absolutely matter as you look at more and more complexity in terms of experiments. Matt highlighted this in terms of the alignment-based insert size of where we have data. Though the key additional feature I wanted to mention is, again, to emphasize these are alignment-based inserts sizes. They are not fragment-based insert sizes, say is something from a tape station or a bioanalyzer. But note the distribution of the sizes, both on the left and right-hand side of the peak. We've been very pleased to see this observation on our surface chemistry that we don't have a very strong bias to the left-hand side of the distribution, meaning that for a given distribution of insert size that we're heavily biased to the short side of the inserts, giving, again, the ability to generate libraries with fairly diverse insert sizes and have those perform very well on the platform. Now, I do also, in all fairness, want to call out, if you had an experiment where you were pooling, let's say the 1,000 base pair peak insert size on the right-hand side and the 300 base pair peak insert size on the far left-hand side of the slide, if you were to pool those in equal molar amounts and put them both on the instrument, we will see there is a bias towards that shorter library during our amplification process. However, we do have some methods to control for that. So if that type of diversity in insert sizes is important, there are some workarounds, both pooling as well as some molecular biology tricks that we know about. So it can be done. It just has to be done quite carefully. So we look at variant calling performance. Matt mentioned some of this already, so I won't belabor it. The point being is, you can primarily decrease this downward. We have additional data sets on this that we are in the process of finalizing, so we won't quite publicly release those. But what I can tell you is with some continued optimization in a couple of different areas that we'll certainly release at the appropriate time, we can see this total error rate actually drop down into the teens. So we're continuing to produce higher and higher quality data on the platform. Matt mentioned the GC context. I won't belabor it other than to say it's quite flat. We've talked about here's just a cherry-picked view of when you reach into those more complex regions with those longer inserts, you do in fact recover meaningful false negatives. These are, again, just a couple of examples shown here. And then finally, wanted to give one quick slide. I won't go through the graphs in detail other than to highlight them. And there's a link to the blog post on Amazon where this came from. This was produced by the team at Scention. Because there's been some very recent discussion as more and more platforms have come on the market that having analysis integrated to a sequencer can result in dramatic whole genome sequencing cost savings. But I think the independent data that's been produced by companies that do analysis for a living like Scention, this doesn't support that conclusion. As you see here, that the on-demand compute costs, and you can pick your favorite platform, whether it's PacBio, HiFi, or Element, or Illumina, are in the less than $2 for the short read platforms and less than $4 for the long read platforms, giving essentially you're talking about whole genome analysis costs that are in a few dollars a sample at the most. and therefore not contributing greatly to the overall cost of these experiments any longer. So, amazing innovation on the bioinformatics front as well. Now, data storage is a little bit of a different question, but of course, those are going to be equal essentially for all platforms, depending on the data density. So we're kind of, leaving those out in the wash for the moment. And if the last couple slides is on some metagenomics and metagenomic assembly data that we recently performed with some partners at Cosmos ID. So we looked at a metagenomic experiment. These were human stool samples where shotgun sequenced and then asked, how do alpha and beta diversity and relative species abundance look on one analysis, and then in a separate analysis, how does creating metagenomic assemblies or MAGs look through on a comparative process? And then so on the left-hand side of this picture, you have 48 to 96 samples, and this depends on the analysis. And on the far right-hand side of that, what looks much less diverse are actually control samples that were used throughout the experiment. So alpha diversity was very, very similar across the board. Here you see both the samples and the controls. But when we went into the assemblies, this is where we saw the effect of some of the higher quality data maintaining assembly qualities, both in terms of total metagenomes assembled. These are binned by completeness, so either 0, 50, 80, 90, or greater than 95% complete. And of course, the number of assemblies that were done were very, go from large to small, depending on the numbers. But note in the lighter blue colors, depending on how much data was put into the system, and then some subsampling efforts. And then whether we used an XT prep, which is the darker blue, or the Element Elevate prep, which is the lighter blue, we see these differential performances, including fairly significant improvements in the mean contig lengths that we saw in the assembly. So same data organization in this case, again, dark blue XT preps, so they were same preps that were used with Illumina, and then the light blue, the Elevate preps. Point being is, although we are compatible with the vast majority or nearly all Illumina-based library preparation kits that are out there, the element-specific library prep kits, obviously we have an opportunity to leverage some of the unique features of our platform. I won't have enough time to go through this in detail, but I did want to mention a few of the applications that we have in our Loop Genomics business, which is a linked long read technology. It's diagrammed here. Importantly, we have the Loop team has been very, very efficient in their publications. And so what I'll do is just kind of scroll through these slides quite quickly, mentioning the ability to do very full-length isoform identification in a variety of samples, in this case, a cancer project. Really saw a significant improvement in the ability to organize and cluster the samples based on their biological characteristics. We can have a very efficient immune repertoire sequencing with the same technology, given that it's a linked long read, so we can stretch across those variable and constant regions in immune repertoire. And then maybe significant for laboratories that do a lot of Sanger, particularly Sanger in very, very high throughput. This is some data that was produced by a customer at Ether Bio, showing that a substantial savings in their large scale Sanger business by using the Loop technology. Delighted to go into more detail with these for both those applications. Full length 16S sequencing, which works exceptionally well, even compared to long read platforms. And then also, again, another example of the Sanger replacement here, which gives some of the economics on the bottom of the slide. What was an $18,000 experiment in a traditional Sanger setting at $4 per read it results in almost a $16,000 savings when run on the Loopseat platform. Now, of course, again, it takes high throughput Sanger to do this, but all these things are possible. Delighted to connect anyone with our team. And then finally, I mentioned metagenomics with a short read, but with our Loop technology, you can also do very, very outstanding meta-transcriptomics, where you're now doing full-length transcriptomics via the Loop technology. We go through a cDNA intermediate and then basically sequence that full-length cDNA via the Loop technology, and it works again exceptionally well for meta-transcriptomics. So with that, I'll conclude this part of the presentation. On behalf of my colleagues here on the call, as well as everyone at Element, thank you all for your attention and joining us today. And I also use this slide as in recognition of the many, many people that have contributed to all of the work that Matt and I showed here behind the scenes at Element that have really made Element a science-first organization and are continuing to drive innovation to make genomics more accessible for all. So with that, we'll have a few minutes for question and answer. Again, thanks for your attention.
Right. Thanks so much, Sean. That was really terrific. And I see we now have lots of questions in the Q&A session. And as a reminder, feel free to enter more questions using that Q&A button at the bottom of your control panel. So we have Matt on, and Sean, you're welcome to come back on as well, because I think some of these questions are going to be for you. The first question is, will the updated Cloudbreak chemistry honor the current reagent cost? And as the marketing person, I can take that one. And the answer is yes, absolutely. It is the same cost as the current kits. Okay, the next question. Why is the flow cell not patterned? Has this anything to do with overloading issues?
I mean, I can maybe address that one. And I think that, patterning is a good technology to be able to control density on a flow cell. It does come with some limitations. I think what we found is we can push the envelope more in terms of density, having unpatterned arrays essentially as they exist today, right? Through some of the gains we've been able to make bioinformatically with spot finding, detecting colonies. There's a lot of innovation in the algorithms there that I think even surpass what we were able to conceive in terms of what densities we'd be able to achieve with those. It's obviously, something that's, on the market today in the AVITI platform and in Cloudbreak, and a place that we still continue to push the envelope. I think we're asking the same questions. Let's go to higher and higher and higher, and let's have the quality maintain, an exceptional level while we go higher.
Fantastic. Let's see, we have a couple questions that I'm going to condense sort of into one here. And it has to do with how to use the cycle kits in different configurations. So for example, does 2x150 mean you can only do 2 paired end reads each at 150? Could you use it as a 1x300? And just in general, does that flexibility exist?
Yeah, I can take that. So the kits are by cycle. There's an additional 34 cycles of reagents in each of the kits to support indexing. Or the use of UMIs, but the flexibility is entirely in the hands of the user, so that either that 150 cycle kit or that 300 cycle kit can be used as a paired 75 or paired 150 respectively, or as a 1x300 as the question asked, or almost any combination therein, meaning the system absolutely supports asymmetric reads. For example, we've seen really exceptional exome data. Instead of doing a paired 150 or a paired 75, if we do 100x50, we actually see really great performance in exome data that way. So there's really no limit in that flexibility, as long as minimum number of cycles are maintained for things like the colony finding, quality calibration, et cetera.
Terrific. What is the performance of AVITI on short DNA fragments between 100 to 133 base pairs?
So I guess I can answer that very briefly. Although there is a circularization process, I saw another question in there that asked how long that turnaround time is. It's an addition-only protocol that takes, it's about 15 minutes of hands-on time, about 70 minutes total. We have a couple versions or modified versions of that protocol for either very high throughput use or to cut a couple of steps out if you want to decrease that time. But we're also releasing an app node on alternative quantitation strategies to avoid qPCR in the loading of the flow cells. So we're really trying to make this workflow as convenient as possible and as low impact as possible. But figured it's a little over an hour is the direct answer.
Awesome. Here's another one. Is the Loopseat Chemistry available to consumers at this time, or is it currently in the R&D phase?
No, Loopseat Chemistry is fully available, both as a service as well as kits. And I do apologize for not finishing the answer on the short DNA fragment question, 100 to 133. The circularization process does not seem to be inhibited by very, very short fragments, meaning we have actually really exceptional data on things that are even smaller than that, down in the microRNA range. So yeah, it works great. The only challenge, and I alluded to this during my talk, is if you were trying to pool exceptionally short fragments with exceptionally long fragments in the same run. Again, it can be done. It's just there are some really important considerations on how to do it well. But a run of very, very short fragments or a run of very, very long fragments perform to the specifications you've seen Matt and I talk about today.
Awesome.
I also, I mean, while we're on the subject of circularization, right? I think I saw in there too a question around, pooling and circularizing, and that is typically what we meant. So one sequencing reaction, you would pool, and then you do a single circularization reaction on that pool. It, of course, can be done on individual libraries that go into that pool. But we find the easiest workflows are, you build your pool, you do the single circularization, and then it's onto the system with enough to do a couple runs from that reaction.
That makes a lot of sense. Here's a question about low complexity. If library complexities are low, do you use sequencing controls to increase the library complexity? For example, Illumina uses phi-X to bring up the diversity.
Yeah, that's a great question. And I think Matt and I might each tackle to each part of this. So with the Elevate chemistry, where we are able to take advantage of a specific sequence during the Polyny finding, so if we use that as the ideal condition, phi-X essentially is optional on the platform, meaning for both low complexity as well as low diversity sequencing, it performs very, very well. We have released some data sets on methylation sequencing where we've shown that phi-X can be used at 2%, at 5%, or 10%. So again, comparatively very low percentages for what is typically a challenging library type, again, being effectively a 3-base genome. Phi-X also remains optional under conditions like that. Now, if you have an Illumina library, a traditional Illumina library, with very low diversity or complexity, then you can, and we recommend using phi-X to bring up the diversity in that situation. But if there's an opportunity to use elements chemistry, you can largely, I won't say exclusively, because there's probably some conditions out there that we wouldn't want to, that we maybe haven't thought of or haven't tested, but you can largely avoid losing some of your reads to phi-X.
Yeah, the only thing I'll add there, Sean, I think is we noticed that last year, right? Nobody likes to throw half their sequencing run away to phi-X to control the bump up diversity. And that was a huge direction that the bioinformatics team here went in, is specifically how do we knock that down? I think we ended up at about a 10-fold less spike required, around 5% needed for low diversity situations to still get reliable data. And it comes down to, how do you identify polonies with multiple colors? How do you, line them up to effectively do, intensity and image extraction from them? And I think some of the gains we made there, purely in the bioinformatics and the software, reduce that amount of phi-X that's needed pretty significantly. I think it's something that we're pretty proud of on the platform.
Fantastic. So Sean, I'm going to put you on the spot as someone who used to run a very large core facility. Here's a question. Any idea as to what level of throughput is needed to make it financially advantageous for a core facility to own the AVITI versus outsourcing the sequencing to an AVITI service provider?
Yeah, so I guess it's a good question, but I won't be able to give you a good answer because obviously Element we don't have any controls over how our current group of core facility labs price their services. But I would say we have both academic as well as commercial companies that are offering services on AVITI. And but as far as the viability goes, I think as Matt indicated, the economics are strong. And I'll just highlight, and without going into the math, but I will say I have a number of models and spreadsheets from my time running a core facilities for 20 years here at Element. If anyone wants to delve into the details separately, I'll be delighted to set up a meeting with you and go through the details. But for the sake of today, I'll just remind that with a relatively low cost instrument, list price of $289 and a service contract cost at $20,000 a year, the amortization of the instrument is pretty attractive, both on a CapEx and an OpEx perspective. And then when you factor in our low reagent costs, it should allow institutions to offer things at a good value. But unfortunately, we just, we don't control how people do that math, right, and how people do that amortization. So it's tough to say exactly when that break-even point would come. But I think, as Matt indicated, we know compared to our competition that we are financially quite a bit advantageous. So, presumably that tip over point would become a bit sooner for us, perhaps, than a competing platform. But I'd be more than happy to go through those numbers with anyone, on a one-on-one basis, that if I could be helpful.
Awesome. So you heard it there. Reach out to Sean. And then I think we have time for one last question. So you show data quality parts that have very high plots that have very high Q scores. Are you planning to announce a Q40 accuracy spec?
Great question. So I guess we've had Q40 as part of our data discussions, and the main reason why we haven't put it out as a spec is the library preparation is such a key factor in that, as is samples. And I think, and this is my own bias, but stated briefly, is every sequencing platform to date has kind of assumed that once you get a sample to library, they're all equal. And that whatever libraries were used to specify a specific quality spec would apply universally to any of those libraries, no matter what the conditions. And we just know that that's not true. And so because of that variability in library through result, we've just been a little bit hesitant to put a wide-ranging Q40 spec. But I think at the same time, we're delighted to put our data up against anyone else's. And in fact, I would also state, as from a scientific perspective, any specifications that are put up without data to back them up and without data to really illustrate under what conditions those were observed, I would view as very preliminary. And I think so those are the reasons behind it. It's not that we're trying to avoid a Q40 spec at all. And in fact, we're actually quite proud of the Q40 data. It's more of, we'd rather underrepresent our capabilities than overrepresent them, as being a new company in the sequencing space and trying to build trust with the field.
Awesome. All right, so that's all the time we have today. Thank you so much to Matt and Sean for sharing your updates on Element technology and applications. And thank you to everyone in the audience for attending. In closing, I will briefly mention we have recorded the webinar and have it available for viewing in the next several days. So keep an eye out for an email with a link to the recording. So conferences that we will be attending, where you can meet us in person by visiting and bookmarking www.elementbiosciences.com/events. Thank you so much for joining us today, and I hope you will join us again at a future webinar. And with that, take care and have a terrific day.